Alexis and Elijah. And Kaya won't be here.
Okay, before we jump into regular class period today, I do want to do a couple of things, follow-up from last week's lab. So one of the purposes of your fermentation experiment was to test two predictions that you made at the beginning of the lab. The first prediction we asked you to speculate on is at which pH will glucose ferment the fastest. And so to get full credit for this, you had to have a prediction, obviously, 3, 5, or 7, and then some kind of valid logic or line of reasoning. I think probably the line of reasoning that was used most often and that made most sense was based upon previous week's lab with the transport and yeast experiment, and one of the conclusions that you should have reached is that yeast have an acidic cytoplasm. Well, if yeast have an acidic cytoplasm, that means that the enzymes that are responsible for metabolism in those cells probably will work best at an acidic environment. And enzymes control the process of fermentation. So the most reasonable kind of defense of that prediction of an acidic cytoplasm would be that those particular fermentative processes would occur best at an acidic pH. Then it asked at pH 5, which sugar do you think will ferment the fastest, sucrose or glucose? Again, we needed a prediction and then some kind of line of reasoning. The line of reasoning that was used most commonly is that people said, well, probably glucose. Elijah, here's your. Probably glucose, because glucose is a monosaccharide and sucrose is a disaccharide, which would mean you would need an additional enzyme, to first hydrolyze the sucrose into glucose, because it's glucose that actually feeds into the fermentative pathway. So, you collected some data, and here's some data from some past lab sections on the effect of pH on glucose fermentation. And so you should have had a multiple series graph for both glucose and fructose with your proper variables on the proper axis, dependent, independent variables, CO2 produced milliliters, time in minutes, and a legend reflecting which of these lines reflected which particular pH. Some of you didn't follow instructions properly and did a single graph of class means. That's not what you were asked to do. You were asked to plot your data on a multiple series graph. So follow your directions carefully. And then a similar graph for sucrose fermentation, again, properly formatted, having a legend. This is a graph of some data that was done in the past, asking whether, or showing sucrose fermentation versus glucose fermentation at pH 5. And here's our glucose fermentation rate for one of the groups, and here's our sucrose fermentation rate. Glucose fermentation rate seems to be higher. But to answer the two questions about at which pH is glucose fermented, and then whether at pH 5 is sucrose or glucose fermented fastest, we need to look at some data and do some statistical analysis. So here's some data from 2011. We pooled 3 lab sections worth of groups into this one spreadsheet. So here is our data for glucose 7, glucose 5, glucose 3, sucrose 7, 5, and 3. And the first thing you should look at is the means, okay, and see what's going on with the means. And so here, glucose 5 has the highest mean, followed by glucose 3, then glucose 7, quite a bit lower, and a similar trend here for sucrose, 5 the highest, 3, and 7. So the first data you would need to look at, and you needed to look at, was this glucose data. At which pH is the sugar fermented the fastest? Can you stop by just looking at this data and say 5 fastest, 7 slowest? No, you can't, okay? You've got to compare and see, is 5 significantly faster than 3? Is it significantly faster than 7? Okay? Then if you're looking at the slowest, you would have to say, is this significantly less than this? Is this significantly less than this? One of the things that might jump out at you a little bit when you start looking at this data is, look at the standard deviation for some of these particular pHs. They're really high. And the higher the standard deviation, the bigger the difference in the means is going to have to be for that difference to be significant. So if you're looking at these right now, you might go, well, that's a pretty big mean difference. But look at all this standard deviation. I would be kind of skeptical just looking at it this way and about whether these differences would be significant or not. Okay? Because the standard deviation is more than half the size of the mean. Okay, so that's a pretty big variation in the data. You look at sucrose over here as well, which we didn't do a lot of analysis of that, but again, pretty high standard deviations. And that's going to come into play when you're comparing glucose 5 with sucrose 5. So again, a difference in means here, but a really high standard deviation, so that difference might not be significant. So you need to go to your t-values and start doing some comparison. Here with an n of 17, degrees of freedom would be 32, n minus 1 plus n minus 1. The 95% expected t-value is 2.042. The 99% is 2.75. So again, these numbers would have to be greater than or equal to this expected value for the difference to be significant at the 95% confidence, or this level at the 99% confidence. And so we start looking at some things. And remember, the one that we were kind of unsure about was, is there a significant difference between glucose 3 and glucose 5? Okay? 5 had a higher mean, but was it significantly different? And you look at this value and you go, no, that does not exceed the expected value for 95% confidence. So 3 and 5 tied. Even though their means were numerically different, there was not a significant difference in these, and so your conclusion would have to be they fermented statistically at an equal rate. And so this data refutes the hypothesis that it ferments faster at 5 than it does 3. Okay? So some of you did parts right, but then you got down to answering those questions about support refute, and you went back to just looking at means and didn't make reference to significant differences and whether those values were really truly significant differences. Okay? And then again, other bottom line is, follow instructions when you're asked to make a graph. Make the graph that you're asked to make and not one that you want to make. Larry, did you have any comments about upcoming stuff for lab this week?
Just be on time. Be ready to go. It's going to be a busy day. One thing I'd like to comment about this, it's important to know how to use the t statistic value in order to determine whether two things are indeed different. That's not just in this class. You will do other things in other courses where that will be important. Also, it gives you an awareness of information that's presented in a statistical manner. For example, right now with the political season, what it is, you get this polling data, and somebody's at 39%, and somebody else is at 35%. But then you look down at the bottom of the table, and it tells you margin of error, which is somewhat similar to standard deviations plus or minus 4. Oh, and you look at that, and you realize it's variation plus or minus 4 around each of those. They're not different. So what you're learning here in simple statistical analysis will help you later on. And I think what this is telling me more so than anything is we got to push this harder than 134.
Okay, so we do have an exam coming up Friday, and it looks like we're pretty much on schedule to finish up the cell respiration information on Tuesday. So that'll give you a break of a couple of days so you're not having new information right the day before the exam. So I will schedule Thursday as a review session for the exam. And so please come equipped with any questions that you might have. I will run over the study guides for these 2 chapters, and if there's some things that we haven't covered in those study guide questions, I'll pull those out and send you an email and alert you to the fact that we might not have covered that question. So be aware of that. So any questions before we dive into cell respiration again? Okay, let's step back a little bit and do a little bit of kind of big picture review. Remember that the purpose of cellular respiration is to harvest the energy that is tied up in the molecular structure of an organic molecule, glucose. Glucose has a lot of hydrogens. Hydrogens have electrons, and that represents potential energy. And that's fortunate because cells need a source of energy to maintain their organization, or else they die. But cells are not set up to use the potential energy of hydrogens or electrons to do work. They need ATP. So that potential energy of a glucose molecule has to be converted into ATP in order for the cell to function properly. So what do? We process glucose through a set of enzymatically catalyzed conversions, one thing being converted into another, until ultimately what we accomplish is we pull off all of the hydrogens from the glucose, and we're left with the carbon skeleton, basically, of glucose, carbon and oxygen, CO2. Okay? Those particular enzymatic reactions, very important enzymes, are the dehydrogenase enzymes, which remove hydrogens from substrates. And dehydrogenase enzymes, to function properly, have to partner with an oxidized coenzyme like NAD or FAD. And as those particular reactions occur, the oxidized form of the coenzyme becomes reduced. And that will result in cellular respiration stopping if the oxidized forms of those coenzymes become depleted, if everything becomes reduced. So we need some way to recycle those coenzymes. In the absence of oxygen, the recycling of the coenzymes takes place after glycolysis, when pyruvate is reduced into some final product of fermentation. In the case of yeast, it's ethanol. In the case of your muscle cells, it's lactic acid. What those reactions will do is pull the hydrogens that were on reduced coenzymes, NAD, pull them back off again and transfer them to pyruvate to produce a final product of fermentation. And that will recycle the coenzymes back to the oxidized state again, thus keeping glycolysis going and giving us our how many ATP that are produced by glycolysis? 2, right? Not a very efficient mechanism. Produces pretty low yields of ATP. Sufficient for some simple organisms, but for complex eukaryotic cells like brain cells, 2 ATP per glucose is not enough to keep those cells going. Fortunately, there is another more efficient form of cellular respiration than fermentation called aerobic cellular respiration. And as long as oxygen's present, pyruvate can do something else. It can enter the mitochondria. Pyruvate still has energy associated with it in the form of hydrogens. So when pyruvate enters the mitochondria, it moves into the matrix of the mitochondria, in here, okay? And there it's exposed to enzymes like dehydrogenase enzymes that remove the remaining hydrogens from pyruvate. And as that happens, we start chopping pyruvate up into smaller pieces, and eventually the 3 carbons of pyruvate become converted into 3 molecules of carbon dioxide, and the hydrogens are attached to coenzymes NAD and FAD. So we start reducing more NAD and FAD. And again, we're going to run out of those oxidized forms pretty quickly. So we're going to need a mechanism to recycle the reduced coenzymes. and that will happen in the electron transport system, which is what we're going to talk about today. And during the operation of that electron transport system, and as those coenzymes have hydrogens removed from them, recycling them back to the oxidized form, we're going to build up a hydrogen ion gradient across the membrane. So here's our membrane. High hydrogen ions out here, low hydrogen ions here. And if there is an ATPase channel protein, those protons, because of the second law of thermodynamics, are going to want to go from their high concentration to their low concentration. And as they do that, that particular exergonic process, spontaneous, high free energy, low free energy, is going to be coupled to the endergonic synthesis of ATP. Now, eventually we're going to reach an equilibrium, right? Hydrogen ions are going to equilibrate. So we need to pump hydrogen ions back out here again to keep this process going. What pumps them? As the electron transport chain is recycling coenzymes from the reduced state back to the oxidized state again, more protons are going to be pumped out here. And then that's going to keep chemiosmosis, the process where we make ATP using the energy of that hydrogen ion gradient. And this is where we get the real payoff of energy. This is where we get the remaining 34 ATP out of that 38 maximum total. Okay? So, what's the correct answer to this question? Why is the citric acid cycle important? Use fingers for response. I see some ones, I see some fours. Any twos or threes? The correct answer is 1. It does complete the oxidation of pyruvate completely to CO2. That is the correct answer. Does it produce ATP? Yes. But does it produce it by chemiosmosis? No, it produces it by substrate level phosphorylation. Does it recycle oxidized NAD back into reduced NAD? Well, no, for a couple of reasons. Number one, that's not how we want to recycle. We want to recycle reduced back to oxidized. But recycling doesn't happen during the citric acid cycle. It happens during the electron transport chain. Or in the absence of oxygen, it happens in fermentation, right? Does the citric acid cycle produce oxygen that's needed by the electron transport system? No. Where does your oxygen come from? Your environment. Remember, cells are open systems. So you inhale oxygen, it's grabbed by hemoglobin in your red blood cells, transported to your tissues, it's dumped off at the cells because the concentration of oxygen is low in the cells. Why? Because they're using it. And so it diffuses into the cells, it diffuses into the mitochondria, and there is used as the final electron acceptor in the electron transport chain. So the only place that oxygen is involved directly in this whole process of respiration is in the final steps of the electron transport chain. Okay, so let's go back and dig into citric acid cycle in just a tiny bit more detail, just to show you some of the rearrangements that are occurring. But again, don't get too hung up on this detail. Always come back to those questions that I said you were going to have to know for the exam about glycolysis, citric acid cycle, and electron transport system. And concentrate on being able to answer the answers to those questions like, is ATP produced? If so, by what process? Is NAD oxidized? Is NAD reduced? Is CO2 produced? Okay? Those should be your really important things you concentrate on for Friday. So, pyruvate is being produced by glycolysis. If no oxygen is present, pyruvate will then be fermented. If oxygen is present, then pyruvate is going to have to be moved into the mitochondria. And there's a transport protein in the outer mitochondrial membrane that will transport pyruvate into the mitochondria. It's going to move into the matrix, and there it will encounter a bunch of enzymes that will process it. We talked about this pyruvate dehydrogenase complex last time. That complicated enzyme that does several things to pyruvate simultaneously, such as remove carbon from it in the form of CO2, remove some hydrogen from it so the NAD becomes reduced, and then temporarily adding a coenzyme onto this molecule, forming acetyl-CoA. So this is the entry product into this citric acid cycle. So when you ask, what does the citric acid cycle begin with? It begins with acetyl-CoA. Acetyl-CoA is picked up by a compound that is part of the citric acid cycle. This particular compound is called oxaloacetate. It has 1, 2, 3, 4 carbons. So if we stick a 2-carbon compound onto this 4-carbon molecule, then this forms the first stable compound of the citric acid cycle. And that particular molecule is called citrate. 1, 2, 3, 4, 5, 6 carbons. Okay? So what we do is we feed 2 acetyl-CoA into this cycle at a time, and those 2 carbons are picked up by the 4 carbons of a compound that the cycle regenerates and produces over and over again. Citrate is modified into an isomer called isocitrate. Isocitrate is converted into alpha-ketoglutarate. This particular molecule has 6 carbons, alpha-ketoglutarate has 5, so we're going to lose a carbon. How? In the form of a CO2 molecule. Remember what those enzymes are called that remove carbon in the form of CO2? Decarboxylase enzymes. This enzyme is also a dehydrogenase enzyme in that it removes some hydrogens from isocitrate to form alpha-ketoglutarate. So where do those hydrogens go? They're picked up by reduced NAD, or by oxidized NAD, reducing the NAD. Alpha-ketoglutarate is converted into a new compound called succinyl-CoA. And again, you don't have to know the names of these compounds, okay? Except probably this one and this one, okay? Because these are really kind of the starting compounds of the citric acid cycle. This is also an ending compound of the cycle, if you like. So 5 carbons here, 4 carbons here. We lose another carbon in the form of CO2. We lose some hydrogen, so more NAD becomes reduced. And again, temporarily, this coenzyme becomes attached to this particular substrate right here. Succinyl-CoA is converted into succinate. That is coupled by a phosphorylation. Technically speaking, what happens is this molecule called GDP is phosphorylated to form GTP. What's G? G stands for guanine diphosphate or guanine triphosphate. It essentially is the equivalent of an ATP, and it can quickly donate its phosphate to ADP to make ATP. So typically when we do an accounting, we say that an ATP is produced here by substrate level phosphorylation. Technically speaking, GTP is produced, but it's quickly converted into ATP. You're going to see GTP later on in the class as an energy source for some important cellular processes, and particularly the process of protein synthesis. That's an energy requiring process. And several steps in protein synthesis are going to use GTP rather than ATP. But as far as you're concerned, think of them as being equivalent. Succinate is oxidized further into fumarate. We don't lose any more carbon, so no more CO2 is produced. But we do lose some hydrogens, and so FAD becomes reduced into FADH2. Fumarate is converted into malate, and then malate is finally oxidized back into oxaloacetate. Lose some more hydrogen, so some more coenzymes become reduced. So for every pyruvate that goes through this process of entering the mitochondria and then going through the citric acid cycle, we produce a reduced NAD up here. Remember converting pyruvate into acetyl-CoA? So we produce one there. We produce another one here, another one here, another one here. So we produce 4 reduced NADs. For every pyruvate that goes through this process, we produce 1 reduced FAD. And we produce an ATP by substrate level phosphorylation. How many pyruvates are produced per glucose? 2, right? So basically, to break down a glucose, this cycle has got to go twice. So we would have to double these numbers. Okay? So what have we made so far at this particular point? We've made 4 ATP by substrate level phosphorylation and glycolysis. But we used up 2, right? We got a net of 2. We produced 2 ATP more. So now our total's up to 4 ATP per glucose. But we've got all these reduced NADs that have been produced. 1 up here in that transition reaction per pyruvate. So 2 there, 2 more there, 2 more there, 2 more there. 8 reduced NADs per glucose. 2 reduced FADs per glucose. We're running out of our coenzymes. And we're also producing 2 reduced NADs from glycolysis, right? We're running out of all those coenzymes. And if we don't do something to get those coenzymes back to their oxidized state again, everything's going to stop. These reactions will stop. The transition reaction will stop. And what will pyruvate do then? It'll go through fermentation. That'll keep it going. Well, these represent potential energy. They've got hydrogens. So it'd really be nice if we had a mechanism to, while we recycle these back to the oxidized form, tap that energy. And that's what the electron transport chain is going to do. So it's going to recycle the energy, but it does so in a very creative way that it traps that energy and basically uses that energy to build up that hydrogen ion gradient we need to make ATP. Now we're at the electron transport system. What's the purpose of the electron transport system? The purpose is to recycle reduced coenzymes. That's what it's there for. Okay? But a real bonus is that as it recycles, it makes ATP. By this process of chemiosmosis. It taps this energy of the hydrogen ion gradient and uses that energy to make ATP. I'll give you a number you're going to have to remember, because when you learn how to do accounting for ATP production, this number is going to be important. For every 1 reduced NAD that is run through this recycling system of the electron transport chain, that can provide enough energy to make 3 ATP by this process of chemiosmosis. For every reduced FAD, that represents enough potential energy to make 2 ATP by chemiosmosis. Now, that's going to be an interesting question. Why the discrepancy? Why does 1 reduced coenzyme give you more ATP potential than another one does? Okay, so we're going to have to account for that. where is the electron transport chain? The electron transport chain is located in this inner membrane of the mitochondria. So when we start modeling this a little bit, I'm going to be kind of using this row of chairs, this part of the inner membrane. I'll tell you why in a minute. Okay? So the electron transport chain would be represented by these people sitting in these chairs. So that's where it's located, in the inner membrane. What it's doing is it's grabbing electrons from reduced NAD and FAD and transferring them to oxygen. And as it does that, it's pumping hydrogen ions out here. What are the electron chain compounds? Well, these are molecules. And they are capable of picking up electrons and becoming reduced, and getting rid of electrons and becoming oxidized. So they're molecules or substances that can become alternately reduced and oxidized. They grab electrons, they give them up. Many of these compounds belong to a class of compounds called cytochromes. Now, not all the electron transport system compounds are cytochromes, but several of them are. What are cytochromes? Cytochromes are proteins, and they have an iron atom attached to them. And it's really that iron atom that becomes oxidized and reduced. It grabs electrons and gives them up. Are you aware of another important protein molecule that has ions that is very important in the functioning of your bodies? Hemoglobin, right? Hemoglobin is a protein consisting of 4 polypeptide chains, and it's got an iron atom in the middle of it. And oxygen can bind to that iron and release it. Oxygen can be oxidized and reduce iron in hemoglobin. Electrons can oxidize and reduce cytochromes during the electron transport chain. So, here is a representation of the electron transport chain. And each one of these circles represents a molecule that is part of that system. And you'll see right here that several of these molecules are the type I just mentioned called the cytochromes. But some of them aren't. There's this molecule called FMN, this Q compound. Okay. These clusters here kind of represent functional groups of those molecules. There's kind of 3 functional groups of those molecules. That number will be important later on. But basically what these molecules do that make up the electron transport chain is grab electrons or hydrogens from NAD. And if we remove the hydrogen from NAD, what happens to it? It's oxidized again. It's recycled. Or they remove hydrogens or electrons from FAD, recycling it. And those are then passed through the system and ultimately donated to oxygen to make water. So when FMN picks up a hydrogen from NAD, it becomes reduced. What if another NAD comes and tries to donate to reduced FMN? Can it take the hydrogen? No, it's already got its hydrogen. It's already reduced. So for it to accept more hydrogens from reduced NAD, FMN has got to get rid of it. So donates it here. This donates it here. All the way down to oxygen. So as long as there's sufficient oxygen present to keep grabbing the electrons that flow through this system, then the system keeps running. But if oxygen becomes depleted, then this compound now is reduced because it can't donate its hydrogens to oxygen. So this one becomes reduced, so does this, so does this, so does this. And so it's like the whole system backs up again. And that prevents these coenzymes from dumping off their hydrogens to the system. This is illustrated in a downhill fashion because this plot represents increasing level of free energy. So basically the electrons are passed downhill from high energy compounds to lower energy compounds. So that's why I like to use this row of chairs to represent the electron transport chain. Higher free energy up here, lower free energy down here. This can help to explain in part the discrepancy between ATP production from reduced NAD versus reduced FAD. This is higher free energy than this is, okay? And therefore has more potential to produce ATP. An analogy to help try to make sense of this. If you've ever seen an old Western movie, one of the things that often happens in a Western movie at a climactic scene is a building burns down. Maybe a saloon or whatever. And oftentimes what the people of the town have to do is they have to form what they call a bucket brigade. And somebody gets at the horse trough where the water is, and at the end of the line there is the burning building, you're throwing the water. The bucket brigade is like the electron transport chain. Okay? And as long as buckets keep being passed from one individual to another, and then that last bucket is thrown on the fire, and then somebody grabs the bucket and takes it back to the horse trough again. As long as you keep that brigade going, it keeps working properly. But if that last person doesn't throw their bucket, they can't grab a bucket from the person next to them. And then they can't get rid of theirs. That is like the electron transport chain backing up as a result of no oxygen present to grab those buckets, to grab those electrons. Okay? So you see how the consequence of something seeming pretty simple, lack of oxygen, can have a real effect on the fact that no oxygen, electron transport chain backs up, coenzymes all become reduced, and therefore citric acid cycle stops, meaning we have to ferment to produce any energy at all. The remaining question, which is a little bit more difficult to understand, is as this is happening, and remember this is within that membrane, why are we, or how are we pumping protons from the matrix out to here? How are we making this gradient? So we've got a high free energy represented by a lot of protons here, and low free energy represented by fewer protons here. What's going on? How is that hydrogen ion gradient made? Well, there's a few things that contribute to this. One of the things that contributes to this is that in the matrix, there's a fair amount of water. Okay, it's kind of like cytoplasm. Like 70% of the matrix is water. And what does water do spontaneously? Water dissociates, right? Into protons and hydroxyl groups. Okay, so that's happening in the matrix all the time. A second thing that's really important, if you look at the electron transport system compounds, there's two kinds of molecules. Some of the molecules donate and accept only electrons. some of the molecules donate or accept hydrogen atoms. So there's two different kinds of molecules: electron donor acceptors and hydrogen donor acceptors. Okay, look at hydrogen up here. What does hydrogen consist of? Atomic number 1, what's that tell you? 1 proton, right? And therefore, how many electrons? 1 electron. So a hydrogen is equal to a proton and an electron, right? So basically, what's the difference between a hydrogen atom and an electron? A proton. Okay, that's going to be important. Third thing, the two types of carriers are asymmetrically distributed. What does asymmetric mean? Remember? Unequal. Okay? Basically what we're saying is they're not lined up in a straight line. So if this group of tiles right here represents the inner membrane, okay, it's a phospholipid bilayer, right? So there's this face that faces the inner membrane space, and there's this face that faces the matrix, right? What asymmetric means is that some of those carriers are located facing this side of the membrane, the side that faces the matrix. Others tend to be located on this side of the membrane, facing the inner membrane space. So they're staggered. Come back to that in a second. So, this is a somewhat confusing, it looks like, slide to begin with. We'll walk you through it here, and then that'll be a good place to... Oh, we need to stop right now. Sorry. Kind of a quiz. This is where we'll pick up Tuesday to wrap up. Okay? So we'll talk about putting this stuff together to make ATP. I'm probably not counting right, so we'll have to make up the difference at the end. Oh, I didn't. Did you guys any at all? Oh, why didn't they have this? Is this one coming down? Or? Did we go down? Pass those down. Okay, if you've got extras, we can share them with the... You're still missing one? You got extras? Anybody have two? 36. Wait a minute. Where did it go? Should have had 2 extras. You've had 2 extras, yeah. Nobody has. Just you got an extra one? Sorry, Caitlin.